Hello, I'm Dennis Neal here at the 5th International Vatican Conference. We're talking today with Dr. Jill Weimer. Now, in medicine, many of these conferences are so specialized, single dimension. How is this conference different, and how is that helpful?
Yes, this is actually my third time participating in this conference, and it's been wonderful to see the evolution that's happened over the last number of years, and how this has brought together leading scientists and really kind of followed the innovation and the pioneering spirit that we're seeing in the genetic medicine, precision medicine, cancer biology space, really to bring together those world thought leaders together under one roof around common goals and common ideas. So I think it's unique because, as you said, there a lot of conferences are really pillared and structured around one topic, whether it's gene therapy or cancer therapy and CAR T. This really brings together folks around the common technologies and the innovation rather than a disease indication per se.
Okay, you were here for a panel on rare diseases. Tell us what you think the potential is or the greatest opportunity for using this genetic targeting to go after a rare disease.
Yeah, so I think one of the exciting things that we've seen in the last couple of years is this idea of applying precision medicine to rare diseases. So rather than taking the approach of one size fits all, copy and paste the wild type gene in to rescue a genetic disorder, really paying attention to that patient's individual genetics and custom designing treatments that really suit them. I think that we know that in the case of particular gene mutations, you can have loss of functions, you can have residual function, and you can have gain of functions all in the same gene that really result in very unique diseases. So, in that case, a gene therapy that just expresses the wild type gene ubiquitously is not going to apply to all forms of the disease. So, looking to technologies like antisense oligos, where we can really target a patient's particular mutation to rescue their phenotype selectively.
Okay, and we always like to end these things on an upbeat note. So, can you tell us some of the more promising new cellular technologies that you're seeing at Sanford Health and elsewhere?
Sure. So I think again, we've really over the last decade focused on singular therapies, looking at the copy and paste of the wild type. I think really the field is evolving at Sanford and beyond to look at how do we actually engineer proteins to get better uptake in cells, to get better targeting to different tissues? How do we look at combinatorial treatments? Are there things that we can actually put together, whether it's an ERT and a lysosomal biogenesis drug to really enhance the phenotype or the correction that we're seeing? So not just focused on sort of that singular treatment and really looking to innovations in the technology and how we screen and test these drugs, right? We've been so fixated on a genetic mouse model that has a mutation in that gene and looking for a correction in the phenotype. How do we actually think outside the box, use unique approaches, whether it's in induced human organoid cultures to be able to screen drugs, use AI approaches and how we analyze those data sets so that we're getting more meaningful data and looking at multiple angles of how we treat these diseases.
We heard a cancer doctor here say that 30 years ago, they were in the dark, they were guessing. And the doctor said, today, we can isolate one gene out of 22,000. Now, if we've come that far, what can you do in the next 10 years?
Yeah, I think it's going to be absolutely amazing how genetic medicine evolves, even in the next 2 years. The steps that we've taken in the last 5 years alone are absolutely unbelievable. Just seeing the number, if you just focus on gene therapy, to go from 2 approved gene therapy treatments on the market right now for rare diseases, I think in the next 2 years you're going to see many more cross the finish line, and it's going to be absolutely amazing to see this, not just from a scientific perspective for me, but from the patient perspective, seeing that they finally, for the first time, have hope, that they're not just at home and told to go and spend the best years with their family because their life will end soon. There's actual hope for these patients that they might be able to live a relatively normal life.
And hope is a great thing to have. And that's what you and the people in your field are giving patients. It's so incredible to think 2 years. Thank you so much for being with us today, Dr. Jill Weimer.
Thank you.